Buenas tardes. Vamos a dar inicio al seminario del doctor Rafael Irizarry. Él es oriundo de Río Piedras, puertorriqueño, graduado de matemáticas del recinto de Río Piedras en 1993. Hizo su PhD en estadística en la Universidad de California en Berkeley. Desde el 2004 es profesor asociado del Departamento de Bioestadística en la Escuela de Salud Pública en Johns Hopkins University. Participó y actualmente todavía desarrolla parte del proyecto Bioconductor, del cual también él en algún momento nos podrá ampliar más información. Ha recibido múltiples premios, el último en el 2004, el ASA Outstanding Statistical Application Award. Además de eso, empezó su gama de premios en 1990, donde ocupó el primer lugar en las Olimpiadas de Matemáticas en Calley. Con esta carta de presentación los dejo entonces con el doctor Rafael Irizarry y su seminario Getting Usable Data from Microarrays.
Gracias. Yo no sé hablar español cuando se trata de ciencia, así es que lo voy a hacer en inglés. Gracias. Thanks to Diana for inviting me and the Department of Biochemistry. I'm going to go ahead and start. So here's an outline of my talk. I'm going to give a quick review of the technology, very quick, of microarray technology for those that aren't familiar with it, so they can follow the rest of the talk. And then I'm going to proceed to talk about data analysis, which is where I'm going to spend most of the talk. And if we have time at the end, I'll mention a couple of things about pre-processing, experimental design, and storing information, but I don't think I'm going to get to that today. So I'm going to be spending most of my time talking about how do you analyze data so that you get usable data from microarrays. So if you have more questions, you can feel free to email me. This is my email address. This is my webpage, but if you Google Irizarry, I'm the first one, so that's easier to find. If you Google Rafael, I'm 15, so you can find me that way too. And this is the project that Diana was talking about, Bioconductor. All the software that I use to make the slides I'm going to be showing today were made using this software. It's free and it's open source, and you're all welcome to use it. It's meant for people who know how to program a little bit, but we can talk some more about that later if you have any questions. Okay, so the first thing I'm going to do, I'm going to spend maybe 5 to 10 minutes talking about how microarrays work, what they're used for. The main application is measuring RNA transcripts. I see this as an audience of biochemistry and molecular biology types, so you all know why that's important, how that could be important in practice. So I won't talk too much about I won't talk about that at all. All I'm going to do is I'm going to say a little bit about how microarrays do this. How do they actually measure RNA transcripts? So the basic idea is to use the hybridization properties of molecules. Okay. So what we want to do basically is count RNA transcripts in a sample. So here's my little cartoon of RNA transcripts rolling around, and this is we know this. We have a gene of interest. We know its sequence. Let's say this is it. So we can construct its complement that will hybridize to this and try to capture these targets on a solid surface. So the basic idea of microarrays is to attach probes, or now we're supposed to be calling them features, according to the organizations that decide what we call things, called MJED. What we do is we attach the complements of the targets we want to measure to the solid surface, and we let them hybridize, and then here we can see that they have all been matched. These are usually labeled somehow, so we can see them with a scanner and count that we have 1, 2, 3, 4 samples, transcripts in this sample. Okay, so that's the basic idea of how they work, what the technology they use to do this. And there's various platforms that you can use to do the same thing. They differ in two basic ways. They differ in many other ways, but there's two main ways they differ. One is the way that the features or the probes, I'm going to be using both words because I haven't gotten used to using features yet. So I'm going to be using features and probes to refer to the molecules that are attached to the solid and target to the sample that has a labeled transcript we want to measure. So one of the way that different platforms differ is by the way that the features are created. One is to sequence them, one way is to sequence them, another way is to use clones. Another way they differ is in the way in which the samples are measured. Some technologies hybridize one sample at a time and use one color in their scanner images, and another technology uses two colors to quantify the amount of RNA. So I have some cartoons to show this. So here's a little cartoon showing the sequenced approach. This is the most popular example are these high density arrays. The most popular company is Affymetrix. Probably the most used technology right now in the United States, I would say. And the idea is to take a little piece of silicone, it's about 1 centimeter squared, and they divide it up. Now they're dividing it up into about 1 million little squares. And on each little square, they'll sequence. So here's a little square, here's a close-up, and we sequence million of molecules on there. We know the exact sequence because we're doing it as we create the arrays. So even a closer look shows the sequence of the molecules on this particular cell. So that's how the sequenced arrays work. And then the spotted arrays usually use robots. Now these days, most of them are using printers that work much better than these robots. And the way at least the robots work, I have a little movie that shows it, is that you have a robotic arm that grabs some of the cloned probes, and it takes some of that and then it drops it or spots them on glass slides. So you can see this happening in this little movie. So that's the robotic arm. It's going to go grab some probe, and now it's going to spot it in various different glass slides, which are going to be eventually identical microarrays. It uses various pins to go a little faster, and you end up with spotted microarrays. Okay, so another way that they differ, so that's, you can either do that first way in which you sequence, that's much more expensive, or another way where you buy the clones and you spot them on the arrays. Two different ways of doing it. Another way you differ is how you hybridize, what you hybridize the arrays. The sequenced arrays are usually one-channel arrays, and I'll tell you a little bit of why that happens in a second. And this cartoon is sort of showing what you do. You have two samples of interest. Say you have cancer and normal cells, and you want to find how the transcripts, the different gene transcripts differ. So you create an array, which I'm representing with these little strands here. These are the features, 4 features on this particular array, and you have the blue gene, the red-green gene, and dark green gene, which are supposed to hybridize to the transcript from the green gene, the red gene, and the blue gene, etc. So then you have one array for each sample. You label them with little suns, and you then let them hybridize, and the blue should stick to the blue, the red should stick to the blue, to the red, etc. And once you have that, you can use a scanner to see how bright everything is. You quantify that brightness, and you end up with numbers, which we'll use for the data analysis. So here we would see that this gene, there were 4 transcripts for this gene as opposed to 0 on this sample, which might mean that this particular gene is of interest. We have another one that's twice as big here than here, and then we have this gene which is just as bright in both, so maybe not of interest, and then there's this green gene wasn't even expressed in our samples. Okay, so that's the basic idea of how the arrays work. And this would be the one-channel arrays. If you want to do this with the 2-channel arrays, I'm sorry, and this is the image you get at the end. So this is a very unrealistic cartoon. You only have 4 features. usually for these types of arrays, you have 500,000 features. So you end up with a picture like this. So you can see all these little squares, right? Each little square is a different cell with a different probe. All right, the other approach is to use two colors. And in this case, we use just one array to compare two samples. So why do they do this instead of the other approach, which seems much simpler? The original reason, and it still seems to be the case, although not as much as before, is that when you use robots to print the arrays, the size of the spots and the quality of spots varies a lot from array to array. So if you took two arrays and you put samples on each one, you might get big differences only because the spots are different and not because the biology is different. So one trick is to balance that by having just one spot to compare the two samples. And then the spot-to-spot variation doesn't affect you anymore, right? Because it's balanced. It's the same for both. So the idea is to use two labels instead of just one to compare the two samples, and then they're gonna compete for the hybridization, but the features have enough molecules to hybridize everything. So that now you have red. One sample is colored red, another sample is colored green. You let them hybridize to the same array, and now you have both colors on the same array, and you use a special scanner that can read them both, and you end up with something like this, where you have, right, so here's only red. Here there's red and green, which averages out to yellow-green. Here there's nothing, which is black, and then green and red turn into yellow. We're using RGB here, not paint. So it turns into yellow. So there it is. That's what it looks like, and we can quantify that and get two numbers for each feature, one for the red channel, one for the green channel, and we can start doing data analysis on that. So here's what those images look like. Usually you get a picture like this. You see now that instead of having little squares that are really close together, you have spots. These are the spots that were set down by the robots, and there's a little bit of distance between each spot, and you can see that some are green, some are yellow, some are red, and then a lot of them are black. But these pictures aren't really that useful to look at. This is not what we look at to come up with results. We usually get the numbers. We get the red intensities, we get the green intensities. So these pictures are nice for the covers of journals, but for data analysis, they're not very useful. Okay, so why am I talking for so long, for 40 minutes, about data analysis? I know that a lot of biochemists and molecular biologists in general that take pride in not needing statistics, right? So you get pictures like this to look at difference in gene expression, and clearly you can see that one of these samples is different from the rest. You don't need to hire a statistician to tell you that. But now, when you do microarrays, this is what you get instead. So what is this picture showing you? Now it's not just one gene on various samples anymore. Now we have, this particular microarray has 12,000 genes. And we have two arrays. We're comparing two samples. So we have, I'm supposed to use the mouse, sorry. We have sample 2, sample 1. It is in the log scale, which I'll say a little more about in a second. And each point represents the expression of a gene on the two, right? So there's two numbers for each point. This is a scatter plot on the two samples. So if we want to, now what we want to know is, just like in this picture, right? We saw testes has highly expressed as opposed to kidney. Now we want to answer that question for 12,000 genes. So one thing we could ask is, which of those 12,000 genes is the most interesting one? What do we look for? Well, the obvious thing to look for in this particular picture is genes that are far away from the diagonal. Right? What is the diagonal? The diagonal are points that are the same on one sample versus the other. And things that are far away from the diagonal are either bigger on sample 2 compared to sample 1, or bigger on sample 1 versus sample 2. Okay, so that's one thing we could do. And now, so what might look interesting are points like this one, which is probably like 5 times as big on one versus the other. This is log base 2. And genes like these that are much bigger on one versus the other. now that I've told you that that's what the interesting thing is, now I'll confess that this that I'm showing you is replicate RNA samples on 2 arrays. So this is all nothing. There should be no expression difference in these two. These should be on the diagonal line. So now we start talking about how do we do it then, when we do have real data. Okay, so here's my view of a microarray experiment, and I have in what is that? Magenta? Magenta, I don't know. In magenta, we have the areas that are where statisticians, biostatisticians, people with experience in data analysis should be involved. And in yellow, I have the parts that you need a lab coat to do. So you start with a biological question, which genes are different on cancer and normal cells, for example. Then we move on to experimental design. What samples am I going to obtain? How many arrays am I going to use? How many samples am I going to use? If it's red and green approach, who gets red, who gets green? All these questions are answered here. Then we do the experiment, and then comes the fun part for me, the data analysis. You get some kind of answer from that you verify biologically, and you interpret, and you're done with the experiment. And while you're doing all this, it's very important to keep the data you're producing, which is no longer small files that you can keep in Excel. It gets a little more complicated. This itself is almost become a science. It's how you keep all this data in a smart way. Also, if you have a big institution where a lot of people are creating arrays, one interesting problem that people are working on is how can you get information from all the experiments that have been kept in a database at the same time. So it's nice to have all the data in one place and have fast access to it for various reasons. And I'm not going to have time to talk about this. All I'm going to say is you should visit www.mged.org and at least read the Miami Standard, which tells you about things you should definitely be keeping, because if you don't, you won't get to publish in Nature and other journals. Okay. So, more about the data analysis part. That was just one little square in the previous slide. It has a lot of little sections in itself. The first one I have there is the image analysis. So the first raw data you get is what computer scientists have TIFF files, which is very simple. You have an image, right? So each little point is called a pixel. Each pixel gets an intensity, a size, right? And now you have to turn this into a table that tells you for each feature how strong was this intensity. So that in itself can be a difficult problem, because remember those little circles? Those little circles were various pixels, not just one. So you have to you have to have a computer go and say, this is a circle. For us humans, it's easy, right? You see a circle and you make a little circle around it. But for a computer, it's not that easy, and we don't want to be making 12,000 circles every time we do an experiment. So you need to have a computer do it. So that is already a relatively difficult data analysis problem. A next step is to do background correction. Something in those cartoons I didn't show you was that it's not just a specific binding that occurs. There's all other things that bind to the probes that are not supposed to bind. So you usually get higher intensities than you should. You need to correct for that. So one of the experiments I've done in Hopkins is I hybridized water to a chip, because I wanted to prove to the biologists I work with that they said it's going to just be 0. So it wasn't 0. It ranged from like 4 to 64, and there was like a normal distribution. We also hybridized yeast DNA to human chips, so that should also be 0 in theory. But there you get a lot of probes that are similar enough that they'll match, and there we got a very, very large distribution. So it is a difficult problem, which I'm not going to talk about here, but it needs to be, you need to deal with it properly. And a second step is normalization, which I will, if I have time, I'll show you a couple of scary slides to make you see that it's important to do correctly. And what this means is, in general, is you want to make data from different slides be comparable. And it's much harder than one thinks. Like I just showed you that slide where I had the same RNA, and you saw this big cloud at the bottom, and that needs to be dealt with. There's other things that can happen. You use a different scanner, then all of a sudden one array has things that are twice as big than in the previous scanner, different days, different temperatures. All these things can affect your results, and you don't need to throw away your data when this happens. You can fix it statistically. It's quite easy to do in many situations. The final step I have here is something I call summarization, and it's what you do when you have multiple features for a gene. So some technologies use 11 to 20 different features for one gene. So now you need to summarize these numbers into one so that you get one measurement per gene instead of 11. You want to simplify things before you go to the more complicated analysis that I'm going to talk about today. That's the last section here. And I have these names here, which I'm going to talk about in a little bit more detail. So by the way, this section here is usually referred to as pre-processing. And this section is usually referred to as analysis. Some people call it high-level analysis, which I don't like very much because I work on this area. So if that's high, that means this is low-level analysis. But I guess that's what I do. Okay. So what are those? What's that high-level analysis part? What is it all about? So these are the three main things people do with microarrays. And one thing I want to stress is that there's some software out there and there's some papers out there that make it seem that whenever you have microarray data, you have to follow this particular methodology, like making a heat map. You know, these pictures. Those of you who are familiar with the literature, where you see the red and green areas, doing clustering analysis. I don't think that's the case. There's very different approaches to these three very different problems. So I'm going to describe the three of them, but I'm only going to talk about the last one in detail, because in academia it's the most popular application. But the fancy journals, the big papers, are usually on the first two. The first papers that appear were doing either class discovery or prediction. So what are these things? So class discovery. This is also called clustering or unsupervised learning. The class discovery, the main idea is that you get many arrays from many different samples, and let's say you have different cancers and you don't know how they're different. You have no idea. So you have an array, you have 12,000 gene expression indexes for each sample, and you have hundreds of samples. And what you want to see is if there's a mathematical thing I can do that will group these things into some kind of class. So that there's a mathematical algorithm that tells me this group of cancer samples are similar, this other group of cancer samples are similar, and these are the genes that are driving it. So you have no idea what's going on, and you just want to find out which cancers are similar. And that's one of the first things that this technology was used for. Also, people want to see which genes move together across samples. That's another type of class discovery. There you're discovering classes of genes as opposed to classes of samples. And there's a paper I like very much on the yeast cycle, where they found many genes that sort of go up and down in a sine wave function, and they discovered that were genes in certain groups that they didn't know about before. Okay, so that's class discovery. There's another popular application which you also need a lot of money to do. It's called class prediction, and a lot of people confuse it with class discovery because it's quite similar, but there's a little subtlety that makes it pretty different in how you analyze it. So in class prediction, you know which group each sample comes from. So you could have two groups, cancer, normal, or this type of cancer and this type of cancer, and you know which is which. And the problem is, in a month, I'm going to get 5 new samples, and I want to be able to predict which group those are in. So it's a little different because I already know the groups here, and it gives me much more information to develop an algorithm. And this is also called classification supervised learning. It's other names for this. So it's a little bit different. There's a lot of papers in in the big journals about this as well, where people say I can predict cancer, this type of cancer, and what they do is they divide their sample at random into two groups. They train an algorithm on one group, and then they can show that they can predict the outcomes of the sample that they left out. All right, the last example which I'm going to talk about, which is the one I'm going to talk about, is class comparison. It's in my view by far the most popular in academia, and I think in part because it's the least expensive of the three by far. And in this case, what you want to know is you have two samples, or maybe 3 or 4, and you want to know specifically which genes are behaving differently in the two samples. Right? So it's much simpler than the other two, or in a way it's simpler, in a way it's more difficult. So you have, say, a very specific type of cell that you maybe treat with some kind of chemical, and you want to see if any genes are expressing differently on one cell versus the other. So you don't know which ones they are, you just want to find out which ones are high here and low here. So it's simpler than than in class discovery, where you're trying to just find genes that are moving together in some nondescript way. Here it's very clear. You want up here, down here, or down here, up here. So this is what I'm going to be talking about. I know already about 2 or 3 people in this school that are working on problems like this, so I hope that they find some of this useful. All right, so the name I use for this problem is differential expression, or detecting differential expression. And the simplest thing you could do is just take 2 samples. Right? So let's say you have a cell and you treat it, and then you have that same cell and you don't treat it, and you get RNA from each one, and you want to use a microarray to measure expression for 12,000 genes. So one thing I can do is I can get 2 Affymetrix arrays, okay? And I put treated cell here, control cell here. I get 12,000 numbers from each one, and I compare those two. That's one thing I can do. Another thing I can do, I can get an adjuvant array, which I think is what you are using. A lot of people are using here. You put red label on one sample, green label on another. You hybridize them to the same chip, and now you're going to get, again, 2 sets of 12,000 measurements, one for the red, one for the green. So they're very similar. It's almost the same thing, except in one you're using one array, in the other you're using 2. But at the end of the day, what you have is 12,000 numbers for treatment cell, 12,000 numbers for control cell. Relatively simple. So let's get back to this picture. So here they are, the 12,000 numbers for this cell, and the 12,000 numbers for this cell. So what am I looking for? I'm looking for genes that are far away from the diagonal. Now, before I continue, I just want to make a very quick remark about using the log scale. There's various reasons for using the log scale instead of the absolute scale. Here's one simple thing to convince you. If I just make a picture, and unfortunately you see this picture in the literature quite often, this is the same picture you saw before without taking the log. This green line down here, 50% of the data, 95% of the data is below that green line. And 50% of the data is below this red line. So that little point there has more data than the rest of the picture. So then the reason is that this data is very multiplicative. So you have, and it has to do with the way intensities are read from scanners and various other factors, but you have a group of things that are between 0 and 100, a group of things that are between 100 and 1,000, another group that's between 1,000 and 10,000. So that's why it's nice to use the log scale, so you get this. All right, so I'm going to say a little bit soon about what the spiking experiment is, but not quite yet. So one more thing about graphs. I rarely ever make scatter plots like this because I got neck problems from making them. Why? Because what am I looking for? I'm looking for genes that are far away from the diagonal. Those are genes that are high on one and not on the other. So what does that mean? What do I have to do? I have to turn my head 45 degrees and look for genes that are far away from. So once I realized that I could instead do this, then I stopped turning my neck. So instead of turning your neck, you can turn the graph. And it has another advantage apart from not getting torticollis, and it's that what you end up with. So this is almost, it's not quite, if there's a mathematician in the room, you're going to know I'm not quite saying what I'm doing, but let's just say I'm doing a 45-degree rotation. It's not quite that, it's very close. And when you do that, what you end up with, and this is just mathematics, when you do a 45-degree rotation, the y-axis now has the difference between the two things. And because we're in the log scale, right, so if you plot x and y and you do a 45-degree rotation, then you have y minus x on the y-axis. Because we're in the log scale, what is that? What is log of y minus log of x? It's the log of y divided by x. That's the log ratio, which is what people usually look at when they want to find differentially expressed genes. So you have the measurement that you care about on the y-axis. That's good. So now if it's big, it's good, it's interesting. If it's close to 0, it's not interesting. So these lines here, these red lines are, M is what I use for log ratio. It comes from minus log of red minus log of green. And then A is for the, I didn't have said this yet, but this is the average. But it's really a pun. It's microarray plot. So that's what people are calling it now. So the M is kind of confusing because it's not very natural, but that's why. You get MA plots, and it's like microarray plot. All right. So the other thing that happens when you take this 45-degree rotation is you get the average of the two log intensities in the x-axis, which is also nice because it means something we care about. Low intensity genes will have small log averages. So if things that aren't expressed will be more towards this way than things that are expressed, which are most towards that way. So we have two very interesting things in this picture: the relative expression and the average overall expression on the x-axis. So what is this red line? That's M equal to 1. This is log base 2, so this is a fold change of 2. If you're above this line or below this line, you have a fold change of 2. Another reason to take the log, by the way, is it makes ratio symmetric. Log of a half is equal to minus log of a half is equal to log of 2. So they'll have the same distance from 0, which is nice. Okay, so anything above these lines will have fold changes bigger than 2. And the first papers that talked about differential expression, that was the cutoff they used. They just said if the fold change is bigger than 2, I'm interested in it. But as we can see here, this is again almost replicate arrays. That's not a good strategy. And now I'm showing you in this picture a little bit more information. This is a very nice experiment that was, it's not cheap, but very nice experiment that Affymetrix ran, and we convinced them to make it public, which has replicate RNA on the 2 arrays. They actually have 60 arrays, but these are just 2. But then they take 16 genes and they spike them in at known differences. So these green points, which I've now denoted with points, have known fold changes of 2. All the other ones, all the little dots, have known fold changes of 1, so they're not differentially expressed. So the ideal picture here would be a line at 0 for all the points, which you know there's always measurement error, so a little bit of scatter, and then these green points should be at 1, which they are close, right? But let's go back to the previous picture. Let's say you just looked at fold change, and you were going to order these points in interest. How interested are you are on these points? Well, the first one would be this one. That has a fold change of 32, 2 to the 5. Then this one is also about 32. Then these are about 16. These are about 8. These are about 4. And these are about 2. So can you see the ones that are actually differentially expressed? It's impossible to tell where they are. There they are. Did you see them before? You didn't. They're impossible to see because you have all this noise. So you would make 1,000 mistakes before you would get to the first correct answer. So just using fold change can be dangerous. That's my first message. The way I think you can fix this is by using better pre-processing, which I won't have time to talk about. This is what that prize I got was about, was how to do this. It was a paper describing it. So this is the same data, exactly the same data, pre-processed in a different way. So you go from this to this. Right? So in this case, we would be able to find at least, at least these guys here. These would be the highest fold changes. And then you have reduced all those things that were way out here have now come down to here. Okay, so you can either you have to take intensity into account for reasons that have to do with how you background correct, and or you can use better pre-processing algorithms. Okay, so that's the first simple application of having to do with differential expression. So you have just two cells, one treated, one not, and you get these values from. But how about what if we want to talk about statistical significance? What about doing inference? Let's say I want to make a statement about the population of cells, or this is cancer samples from humans, about the population of humans with cancer versus the population of humans without cancer. So in this case, just like everything else we do, you want to sample from the population. So let's say we wanted to make a general statement about these cells. You would want to have a random sample of, say, 5 to 10 cells, depending how precise you want your answers to be, and compare them to 5 other normal cells, and look at the variation within these groups to see if the changes we see are real or just due to chance, right? Because now I'm introducing a new kind of variation, not the technical variation that we saw that had to do with the technology and how you pre-process the data, but biology, right? You can have the same gene on the same exact types of cells behaving very different just because nature is random. So we want to be able to see that variation and then compare the changes we see to that variation and be able to do inference as we usually do. So we want to, if we want to be doing that, we want to do biological replicates, which will permit us to do inference about a population. I always advise the people I work with to never do technical replicates because they only permit inference about the sample you are looking at, not the general. You can't make general statements. So technical replicates are useful to test your technology maybe once or twice a month. But for the experiment itself, I recommend never wasting money on them and going straight to biological replicates, which contain the same information as technical replicates and more, because it also includes information about biological variation, which is very important to know. So now the next question is, what test do we use? We have now various, for each gene, which before we had 2 numbers, control and treatment. Now we're going to have, say, 3 controls and 3 treatments. So now how do we put those numbers together to talk about significance or to give some summary statistic? Do we average the fold changes? Do we do a t-test? Do we Wilcoxon test? What do we do? So, right, so that's the next question I'm going to talk about. So the first thing I'm going to do is I'm going to talk about exploratory data analysis. That's what, when statisticians write EDA, that's what they mean. Other people mean other things. It's just looking at your data before you look at the final numbers. So let's look at the most obvious thing. First, you have 3, for each gene, you have 3 controls and 3 treatments. What is the most basic thing we could look at? Well, we could take the log. Well, we're always taking the log, so we have the log intensities for these 3, the log intensities for these 3. We could take the average and then take the difference of the average, which would give us an average log fold change. So this is sort of an average relative expression between the two cells. So we could make a picture of that. Now I want to make a quick comment about the overuse of heat maps. Some people will do heat maps and say they make this picture to try to find that their samples are different, and I'm not going to harp too much on this, but that's not really what we want to be looking at when what we care about is finding a few genes that are different in the two samples. When we do this, what we're really looking for is to see if the samples automatically divide into two groups, which in this case they don't, and they shouldn't, because it's only 16 genes out of the 12,000 that are different. So instead, what I'm going to do to start is, again, make an M versus A plot, but now it's averaged, because we have 6, not just 2 numbers. So instead of plotting the difference against the average, I'm going to be plotting the difference of the averages between the two populations against the average of the averages between the two populations. So it's going to look like this, and this is using that better algorithm, this better pre-processing algorithm. So I'm plotting what I care about, the average fold change on the y-axis and the average of the average intensities on the x-axis. And again, what I see, I can look for points that are high, and those I can consider interesting. Okay, so now the next question is, that just looks at the differences in averages. But what about variance? So do I think that different genes have different variances within replicate populations? So if I take a bunch of cells that should be the same, they're all genetically the same, do some genes behave differently just because nature is that way? So this picture comes from an experiment that I participated in, where the original purpose of this experiment was to decide if pooling is a good idea or not. But there's something else that comes out of it, and it's how much does the variance of genes vary across genes. So let's just look at the right panes, because this one has to do with pooling, which I won't have time to talk about. Let's just talk about this one, and this one, okay? Don't look at the left pane. So we're looking at 1, 2, 3. All right, so this experiment has 2 groups of mice. One has been treated, one hasn't. And I have 12 mice. For each mouse, I have an array. So in this picture, I have 12 green dots. There's 10 dots all lumped together down here. Those are the expression for gene number 1 on the 12 mice in the control group. And over here, where's the mouse? Okay. Here I have the 12 treated mice, their expression for gene 1. So I chose these 3 genes because they all have about the same average log fold change. So on average, these 3 genes would be ranked in the same way because their fold changes on average are about the same. They're about between 2 and 3. So let's look at the first one. Here's the average of the first group, and down here is the average of the second group. So that's a fold change of about 2, maybe a little more. It's more like 3. Okay, so here's 50, 250, and this is log, so 100 is about here. So yeah, so that's about twice as big, a little more. So if I just look at fold change, I would say this is a fold change of 2. That's pretty interesting. Now let's move to this one. Same thing. The average is about here, and the other average is about here. So that's also a fold change about 2. And then the third group, one average here, the other here. That's about 2 as well. Let's look at this group. This difference is clearly occurring because of 3 outliers. So that shouldn't be interesting. So there seems like we should be looking at something other than just the average fold change. Similar here, this group is much tighter than this one. So what is it that we need to be looking at? Well, it's the gene-specific variance. This group has a bigger variance than this one, so maybe it's not as interesting. This is a much tighter group than this, so the difference would be considered more statistically significant. So this is something you all are used to. You've done this before. And we're going to just do it in the same way. The only difference is instead of having one thing, we have 12,000 things we're doing at once.
What is the red plus on your first panel?
Say that again?
The red plus, gene 1.
Yeah, that's the additive average. I'm not going to have time to talk about that particular issue. That's another reason for taking logs. Outliers are less prone to affect your averages.
This was on inbred mice.
These are inbred mice. Yeah. So these are all twins, and you can see there's a lot of variance for that gene, the expression of that gene. All right, so there's a lot of, one of the reasons I'm showing these relatively simple examples is because I think that biologists as well as statisticians are intimidated by the literature in microarrays, because there's all these fancy methods with fancy names, but a lot of them are just doing very simple things if you look behind the hood, under the hood. And I want to, those of you who are inclined to quantitative fields, you don't want to be afraid. You want to just try to figure out exactly what they're doing and realize that it's not that complicated. So I'm going to show you one very simple thing we could do that leads to a lot of the fancy things that other people have done in a very simple way. So what do biochemists do when they have 3 numbers and 3 numbers? Right? They usually just do a t-statistic or a t-test to see if it's statistically significant or not. So let's just start doing that. So the t-statistic has various nice properties. One of them being that it's in certain circumstances it's t-distributed, and it has a nice interpretation because it's the across group variance divided by the within group variance. So it sort of tells you how big is this difference compared to how much the thing vary when it shouldn't vary. So, and that's why it's used in many cases for defining if something is significant or not. So there's a new plot I'm gonna show you. It's called a volcano plot, and all it does is it plots so you do a t-test and then you get a p-value. So I'm gonna plot the p-value on the y-axis against the m, so the average log fold change in the x-axis. So here's what these two look like. So in this picture, this is the m, the average fold change, which is now on this axis. And in the y-axis, I have the p-value in the log scale, and it's going up as the p-value gets smaller, right? Because small p-values are more interesting, so I turn it around so that up is interesting. So I guess it looks like a volcano. I really have never seen it, but that's what we call it. Maybe it's the eruption, I don't know. So this picture is very useful because one of the things it shows you is that if you only look at the p-value, and let's say you consider anything that has a p less than 0.01 to be interesting, look at all these points here. These are about 50 genes that have p-values less than 0.01, but their average fold change is very small. So how can the p-value be small when the size of the difference is small as well? Well, it must be that the denominator in the t-test, the variance, is also very, very small. So this can also happen by chance. So we can get p-values that are very small by chance as well. And in this picture is kind of showing us that you shouldn't just look at p-values either, because they can be misleading as well. And one thing that I like to do is I like to look at both things at once. So in this case, I would say, show me genes that have big fold changes and small p-values. And that's what this group is here. This group. And it turns out that this is from the spiking experiment again, where 16 genes were truly different and the rest weren't. So all these are false positives. These guys are all true positives except these 2 points. So I wouldn't do that bad if I made a picture like this and kind of picked these genes because they look kind of different. All right, so here's another picture using that other pre-processing algorithm that's a little noisier. And what this shows you is that when you use a bad pre-processing algorithm, the t-test helps quite a bit because a lot of genes that had big fold changes are given big p-values, so they're not considered interesting because they were big only because they had big variance. So those go away. So that in this case, it helps. All right, so you don't necessarily have to read through all this. What I'm going to say is quite simple. So why the t-test was doing with the comparison of 3 versus 3 is actually quite bad, and the reason is that you do a very poor job of estimating variability when you only look at 3 numbers. So why is that? Because you have a big chance of making the denominator very small when it really shouldn't be that small. And then you get a very big t statistic which gives you a very small p-value. So there's been a lot of work by groups of statisticians, computer scientists, other data analysts to improve the t-test in certain ways, and they've come up with these things that a lot of people are calling moderated t-test. And I have some examples here. One very popular one comes from this procedure called SAM. It's very popular one because it appeared in the PNAS and also because I have an Excel add-on, unlike many of the other things I have here. And I'm not going to go through all the details here, but I want to describe really quickly what SAM is so that you can see how simple the idea is and why it works. So I'm using T to describe the average log expression in the treatment group, C to describe the average log expression in the control group, and S is the estimate of the standard error. So the average log fold change is simply T minus C, right? The average in the treatment minus the average in the control. That's fine. One of the problems with this statistic is it doesn't incorporate the gene-specific variance. So if a gene has a lot of variance, this could be big just by chance. The T statistic divides by something that's trying to describe the within group variance. So T minus C divided by S is the t-test. And the problem with that statistic is that when you only have 3 versus 3, I use that example because half my collaborators, that's how much money they have, 3 versus 3 money. That has the problem that S can get small by chance, and then it blows this ratio up artificially. So what does SAM do? SAM says, well, let's add a constant to the denominator. So I put a little 0 there to denote that this constant doesn't change from gene to gene. So what that does is it says this denominator can never be smaller than S0, S0. So it can never blow up. And they have a trick for finding that S0, which works nicely. And what you end up is with a moderated t statistic. It doesn't blow up. It's very ad hoc. So statisticians that are bothered by ad hocery developed a Bayesian or empirical Bayesian approach. If you don't know what that means, don't worry. But they came up with almost the same answer using formal arguments, which is nice to know. Wilcoxon is a robust test to outliers. It only uses the ranks, and it works well. But with 3 versus 3, getting 3 things that are bigger than another 3 things is not that unlikely. So it's not going to be very powerful in a case of 3 versus 3. Well, I have some notes here of where you can get these tests. This is if you're using Bioconductor. If you're using commercial software, I can try to help you find it. Or at the end, I can answer some questions because most commercial softwares these days have some of these tests incorporated. All right. This picture I'm not going to talk about. It's only showing you that there's a big difference on a 3 versus 3 experiment between SAM, T, and fold change. In a 3 versus 3 example, SAM works best, or things like SAM. T works worse by far, and the fold change is kind of in between. But that changes when you do 12 versus 12. When you do 12 versus 12, the t-test is fine. You now have the power to estimate the variance, so you don't have that problem anymore. And t-test, SAM, Wilcoxon, they all work the same. All right, so the last thing I wanted to talk about is hypothesis testing. So now we have a statistic that we can use to rank genes. So the bigger the statistic, or it's a p-value, the smaller the statistic, the more interesting I think the gene is. But what if I want to throw a cutoff and say genes above this cutoff that have the statistic bigger than this cutoff, or the p-value smaller than this cutoff, are significant in some way? Or I want to publish a paper in a journal and I just have a short list of genes. That's a tricky step. The main problem with this is the meaning of p-value. If you get a p-value for each gene, and let's say it's very small for all the genes you pick, you make a list of 100 and they all have very small p-values. Those p-values do not mean what they used to mean, right? When we do a very simple experiment that we have one thing and we have two groups, we get a p-value that we know what that means. Under the null hypothesis, what's the chance of seeing a difference this big? That's what a p-value means. But now, everything changes because we're no longer asking what's the chance of seeing a difference this big given the null, because that's not what we're doing. What we're doing is we're looking at 12,000 differences, picking the biggest 10 or the biggest 100, and then asking how likely is that. So just to give you an idea of how different that is, if you fill up a stadium with 10,000 people and you ask everybody in the audience to toss a coin 10 times, 0.05, well, I'm talking about 0.001, there's going to be 100 people that are going to toss 10 heads in a row on average. So that has a p-value of 0.00, well, 1 divided by 1,024, so about 0.001. Is the person that tossed 10 heads really special? No, they weren't. They were just really lucky. And because we had 10,000 people, we happened to get 100 people that did it. So in a more general context, if you have 10,000 genes and the null hypothesis is true for all of them, so nothing, you're just comparing replicates, nothing is different, you will, on average, get 500 genes with p-values of 0.05 or smaller. That's going to happen if you did things right. So p of 0.05 does not mean what it used to. That is something you have to keep in mind. They have a name for this. It's called the multiple comparison problems. Statisticians fight about how to fix it, but there's no fight about the first 4 points. There's no fight that it's a problem that you can't just report p-values. So you can use p-values to rank or to just get an idea of how interesting things are, but you shouldn't interpret them like you used to. There's other approaches. One popular thing is something called the FDR. This stands for it's not Franklin Delano Roosevelt, it's the false discovery rate. And basically what it tries to estimate is in a list, let's say you give a list, it tries to estimate how many mistakes you made in that list. So you give a list of 100 genes and you say, here's my 100 genes that are interesting, and I think that about 5% of them are wrong. So you might ask, but wait, why not make a smaller list with 0 mistakes? The list with 0 mistakes may have 0 things in it. So you won't publish a paper with that list. So there's other approaches. Again, I said there's fights about this. I'm telling you this one because it's the most popular, as far as I can tell. Okay, so I'm going to stop here, and I'm going to take questions from anybody who wants to ask questions. Okay.
Gracias a Rafael por la presentación. Si tienen preguntas, por favor, acercarse al micrófono para que quede en la grabación. Gracias.
How complicated it gets when you're using human samples and you have polymorphisms in the genes?
Complicated. So let's see, how can I answer that? What chips are you using? Well, I can answer it for both cases. So Affymetrix, you've heard of this company? Yeah, okay. So Affymetrix, and there's other companies, NimbleGen is another one. they'll choose relatively small probes from the genome, and they'll try as best they can to avoid regions that have polymorphisms. Right? So in that case, it shouldn't make a difference. But I'm sure they make mistakes. Right? But that's one way that you can deal with. Nimblegen will make any chip you want. You can tell them what sequences you want, and they'll make it. So if you know where things you're worried about are, you can ask them to exclude them. Also, you can, if you have someone that's very relatively good with computers, you can have them take a chip that's commercial and remove the probes that you're worried about because of polymorphisms. But yes, that's an issue, and it hasn't been looked at very carefully. Yeah?
De una gráfica de MA, ¿cómo tú sabes cuándo tuviste una buena data o no?
Well, I can tell you when you know you didn't get a good data. It's hard to tell. So, ¿quieren que lo conteste en español o en inglés? No te importa. Déjame enseñarte una mala. So this is a bad MA plot because it has a curve in it, right? So you have, for these genes, they're all at -1, and then it goes up to 0. So you certainly do not want to see curves. So something you can do is, if you think you have a good lab setup, you could look at all MA plots in your experiment. You have many hybridization, and you start getting used to you yourself. This is very lab dependent. This is why I can't just tell you. Different technicians are going to have different widths. I have a guy that has very tight widths, and I also work with people that have very big widths. But you know, that's just variance. You can deal with it by averaging things and whatnot. So, but you're more concerned about quality control. So one nice thing to do is, if you look at an MA plot. Are you using 2-color? So if you have 2-color, it's easier. You just make you make an MA plot. Every experiment, you look at an MA plot, and you have experience, so you know what your typical MA plot looks like, and you'll be able to see the bad ones. So one statistic you could look at is, first, you have to make a picture to see if there's curves or not. Another thing you can look at is the variance, this variance. So you take all your M's, and you see, like, you take like the inner quartile range or something like that, and you get an idea of how big that should be. So whenever you see something much bigger than that, then you might worry. There's other things that are bad, as if you see a lot, like a line here, that means you saturated the scanner, because there's all these things get really big, and they just go against 16. It's 2 to the 16, and then they form a line in the MA plot. So there's various things like that you can do. But it's very lab dependent, it's very technology dependent. If you have a lot of data, you'll be the expert, and you'll be able to answer that question, just looking at a lot of them. Yeah?
Trabajando en casos con pacientes y controles de cáncer, si uno quiere establecer los controles y no quiere hacer pooling, sino que lo va a estar diferente, como cuántos controles uno debe establecer para un tipo de cáncer en particular, y controlando por ethnicity.
So you're going to use 2 colors, and you're going to use a reference sample, and you want to pool 2 color, and you want to have one reference sample that you always use. Yeah, that's tricky. Everything is easier if you match. Can you do match? Okay, so if no. Okay, there's two different kinds of pooling. You do not want to pool your interesting things. So the treatment group, or in your case, the disease group, that you don't want to pool. The reference, sure, you can pool. So you want to have a reference that's common to everybody, and I don't think it's as important to make that be like very. to represent the entire population, what you want to make sure is if you make a big batch once and you always use the same batch. That's the most important thing for references. Because you don't want, what happens if you use a different control in two different treatment groups? Then you might get differences because the controls are different, not because the treatments are different. So if you're going to use a reference, I don't think it's the details of what that reference is as important as using the same exact thing every time. That's what's important. And if you can match, that's better. Okay. Yeah. So yeah, in some of these newer arrays, like if you use Affymetrix, you wouldn't need a reference, right? You just do one. You do everybody. Yeah. Okay. But if you use one channel, so let's say you say forget the pool, let's just do one channel, then you don't need to have a reference that way. You can just compare them to each other. It depends what you're trying to do. You're trying to find genes that are different in cancer as opposed to the normal population. Yeah. So yeah, I would make a pool that represents the general population and always use the same one. And be very certain that it's working because everything's going to depend on that. It's a hard one.
Y en ese caso, el pool puede ser de 3, como hablaste. O sea, si yo tengo una referencia, pueden ser 3 cultivos distintos, lo uso como referencia y lo comparo ahora contra mis muestras.
Okay, this all depends on the question you're asking. If you're, I have a little picture here. So let's say this is one example. If you're comparing A versus B, some people will always want to use a reference. Okay, so you're comparing, you have A on the reference and B on the reference. And all you care about is the comparison between A and B. If that's what you're doing, you don't necessarily need a reference because you can compare A to B directly. And you'll reduce the variance quite a bit. So that's what I try to do. I try to avoid having to use a reference when I can. So if you have an experiment where you're doing mice, you're usually comparing treated and control, or then you just go ahead and you compare them directly. Let's say you have 3 mice control, three that aren't. You randomly match them and you do it like that. That's, I think, better than creating a pool and so you don't waste more arrays. It's
addressing that same question of variance between biological replicates for the same gene, right? So when you're making a decision about how many to do,
how many arrays to do?
Yeah, is that a matter of experience? You know, if in your experience you're getting a lot of variance from a certain group of genes, which means that they're really not changing, they're just, nature is that way, as you said, then how many biological replicates is enough?
Yeah, so that's a hard.
It's a minimum of 3.
That's a hard question. No, there's no minimum, there's no maximums. A minimum of 1 and a maximum of as many as you can afford. So
the more the better.
The more the better, but yeah, I mean no, because you waste it's money, right? So yeah, so I usually I don't like these types of questions because they're very hard to answer, and it usually comes down to everybody knows the real answer is money divided by price, and if that's what it is, then why bother thinking about it? But let's say you actually do have to think about it. Here's how it's tricky. Let's think about what a typical power calculation is. A typical power calculation is you have 2 groups, and you care about observing a difference in the average of those 2 groups, some size, and you know that each group has a certain variance. So you can say I'm going to do a p-value of 0.05, and I want to know what the power is. So what is the chance of not seeing this difference when it's really there, right? That's a typical power calculation. Now let's think about a micro experiment. Now we're doing 12,000 genes. So what is the question? Is the question, I expect 100 genes to have this size of a change, Or is it, I know they're all changing and I want to find the most interesting ones? So you see how the question, just the question itself is completely different. So right there, it's much harder. And the other problem is, now each gene has a different variance, and I don't know what it is. Maybe I have an idea of what those variances could be. So if you have the only way to answer this question is you write down specifically what you want to know. So and that question could be very strange. It could be, let's say I think everything is differentially expressed, but I want to find the 100 most differentially expressed. So my power may be, I want my list of 100 to contain 25 that are in the top 100. See, that doing the math for that is impossible. You actually have to do a simulation, and in that simulation, you want to make it realistic. So you want to make each gene have a different variance, and how you pick those variances, I have tricks for doing that, but it has to do with observing, doing an experiment where you only observe the biological variance to be able to imitate it in a power calculation. So it is a very, very hard question, and don't believe anything you read about 3 being a maximum or a minimum, because it depends on the experiment, it depends on the lab, it depends on the organism, the tissue, everything has an effect here.
Gracias nuevamente a Rafael y a ustedes por su asistencia.